All right, guys, we're going to go ahead and get started. Good morning or afternoon for many of you, and welcome to the Sekisui Xenotech's webinar, Evaluation of Ketoconazole and its Alternative Clinical CYP3A4-5 Inhibitors as Inhibitors of Drug Transporters. The in vitro effects of ketoconazole, ritonavir, clarithromycin, and itraconazole on 13 clinically relevant drug transporters. My name is Matt Beck, and I'm the Global Marketing Manager here at Sekisui Xenotech. Today I am pleased to be joined by Dr. Lydia Vermeer from our Drug Transport Group. Before we get started with today's presentation, I would like to go over a few housekeeping items. Here is an example of your go-to webinar user console. Using the orange arrow will allow you to minimize and maximize the user console. Please note that your audio is muted. If you have questions during the presentation, please post your questions in the questions pane seen highlighted here. We will have a Q&A session immediately following today's presentation to address questions posted via the questions pane. A copy of the slides and recorded session will be available a few days after the presentation. We will send both links via email for your reference. It's important to note that we send these to the email address that you registered with when you registered for the webinar. So if the email address is either incorrect or missing a letter, there's a chance you may not get it. If that happens, please email me directly, and I will get you a copy of the slides in the webinar. At the conclusion of today's webinar, a short survey will populate. Please take a minute to fill it out, as it helps us to gauge how we did today and also allows us to be a resource to you in the future. We truly do value your feedback. I am pleased to have Dr. Lydia Vermeer here with us today. Lydia received her Ph.D. from the Department of Medicinal Chemistry and Natural Products at the University of Iowa, where she studied neurotoxicology. After completing her degree, she held a postdoctoral training position at the University of Kansas Medical Center, where she used a novel rodent behavioral model to study migraines and the effects of environmental estrogen exposure. She joined Xenotech in 2014 as part of the Drug Transport Group, where she focuses on managing multiple client protocols and researching the development of novel assays to assess drug transport activity in the hopes of better understanding the impact of drug transporters in vivo. Lydia has published several peer-reviewed journal articles and is a member of scientific societies such as SOT, OSSD, and SFN. It is my pleasure to present to you Dr. Lydia Vermeer.
Thanks for that introduction, Matt. So before we get started with the information we can have in the paper, I'm just going to go a little bit over the drug transport services that we offer at Sekisui Xenotech. We have all the major SLC or uptake as well as ABC or efflux transporters. On the uptake side, we have OAT 1 and 3, OCT 1 and 2, and OATP 1B1 and 1B3. We're utilizing a stably transfected HEC293 test system in these, and we also have the wild type cells. On the efflux side, we have MDR1, also known as PGP, and BCRP in our MDCK2 transfected cell lines. We also can test Keiko permeability as well as PGP inhibition on our Keiko2 cells. We also have MRP2, 3, and 4, and BSEP in our membrane vesicle test system. PGP and BCRP are also available in the membrane vesicle system. When we design a protocol for our clients, we utilize the FDA and EMA guidance documents. They recommend the 7 major transporters, which include PGP, BCRP, OATP 1B1 and 1B3, OAT 1 and 3, and OCT2. And the EMA also recommends looking at BSEP. They ask you to consider the transporters including MATE1 and MATE2K as well as MRP2. Both require that you test all 7 of the major transporters for inhibition. And then when it comes to substrate evaluation, typically PGP and BCRP are recommended, and the others are tested based on their clearance or elimination routes. So for example, if your compound is cleared over 25% renally, we would likely recommend testing OAT1, OAT3, and OCT2. So getting into the paper, as a bit of background on ketoconazole, this is an orally available drug. It's a synthetic broad-spectrum antifungal agent of the imidazole class, as you can see from the figure at the bottom of the slide. In 1982, the FDA approved its use for fungal infections, and since then it's known to be a substrate and an inhibitor of CYP3A4 and CYP3A5. In the past, a high dose of ketoconazole was previously used as the gold standard for a strong CYP3A inhibitor in clinical drug-drug interaction or DDI studies. In 2013, though, the FDA banned the use of ketoconazole from the clinic, and this was due to asymptomatic reversible liver function test abnormalities. Basically, as soon as the compound was approved for clinical use, people began studying the potential for DILI, or drug-induced liver injury. And we can see that as early as 1984, Van Tuyl estimated that between 0.1 and 1% of patients could have DILI occurring following treatment with ketoconazole. While there was no association with dosage, as you increase the duration, the possibility of DILI increased as well. In later estimates, we found anywhere from 134 per 100,000 cases, or upwards of 3.6 to 4.2% of patients that exhibited these asymptomatic liver function abnormalities. When the FDA banned ketoconazole from the use in the clinic, they specifically recommended clarithromycin or itraconazole as an alternative for DDI studies, but they did note that other drugs may be used. Ritonavir has been suggested as an alternative CYP3A inhibitor by some, including Greenblatt, but Alice Key's paper excluded this due to nonspecific cytochrome P450 inhibition as well as induction. Diving into that paper a little bit more carefully, we can see that they systematically tested 19 strong CYP3A inhibitors. They found that only itraconazole and clarithromycin were acceptable. They excluded others for a variety of reasons, including that the drug is not approved in the United States. It's known to be a nonspecific cytochrome P450 inhibitor. It might have significant safety issues in vivo, potentially exclusively used in combination with ritonavir, which would make it less useful with other classes of drugs, or it was only a moderate CYP3A inhibitor. So in the literature right now, there is some transporter data available in terms of inhibition by these compounds, but previously there's been no comprehensive investigation of alternative CYP3A inhibitors as transporter inhibitors as well. The information that we can find includes ketoconazole IC50 values for PGP inhibition that can vary 37-fold. Over 6-fold variation can be found for OATP1B1 inhibition. For ritonavir, we saw over 80-fold variation in PGP inhibition and over 117-fold for OATP1B1 IC50 values. So the goal of this study was to define the inhibition profiles of these compounds listed here: clarithromycin, ritonavir, and itraconazole, along with 3 itraconazole metabolites: hydroxy-itraconazole, keto-itraconazole, and N-desalkyl-itraconazole. We tested these in comparison to ketoconazole, and we were looking for an inhibition profile towards these transporters, including OATP1B1 and 1B3, OAT1 and 3, OCT1 and 2, MATE1 and MATE2K, PGP, BCRP, MRP2 and 3, and BSEP. And the overall goal was to allow for a more informed choice of a strong clinical CYP3A inhibitor in clinical drug-drug interaction studies if you have a drug candidate that you know to be a substrate of one or more of these drug transporters. Because so many things are happening in vivo, typically you can have metabolism by CYP3A as well as drug transport that can affect the clearance of a drug. So what we're trying to do is reduce confounding DDI results. The publication details are shown here. The paper was published in the March 2016 edition of Drug Metabolism and Disposition. My colleague Caleb Isringhausen and myself carried out the assays and processed the data. Brian Ogilvie and David Buckley assisted in data interpretation and did the majority of the manuscript writing. I've shown here the test systems that we used in these assays. As I mentioned earlier, we have a transfected HEC293 system. These are immortalized cell lines, and we had a stable transfection for OATP1B1, 1B3, OAT1 and 3, and OCT1 and 2. We also again had the control wild-type cells for HEC293. We had transiently transfected cells for MATE1 and MATE2K. All HEC293 cells were seeded and cultured for 24 hours. Following that, OATP1B1, 1B3, MATE1, and MATE2K were supplemented with 2 millimolar sodium butyrate. The rest of the cells were fed with fresh supplemented DMEM, and all cells were cultured for a further 24 hours before we used them in assays. For the vesicle assays, which include PGP, BCRP, MRP2 and 3, and BSEP, we are using an inverted membrane vesicle system. This is an ATP-dependent test system in order to have a functioning transporter. We used industry and literature standard substrates for all of our transporters, which include estradiol glucuronide for the OATPs and the MRPs. p-aminohippurate for the OAT1, estrone sulfate for OAT3 and BCRP, TEA for OCT1, metformin for OCT2 and both mates, and TCA for BSEP. These were all radio-labeled compounds, and we used liquid scintillation counting in our analysis. Finally, for PGP, we had cold NMQ, or N-methylquinidine, which we utilized our analytical department here at Sekisui Xenotech for those samples. All inhibitors were pre-incubated with our HEC293 cells, or the vesicles, for 15 minutes prior to the addition of our substrates. We were trying to minimize the effects of time-dependent inhibition as well as non-specific binding of the compounds. We used a substrate concentration of between 1 and 10% of the Km for each assay, and this was so that our IC50 approximately equaled our Ki. This table here indicates all of the concentrations that we used for the different uptake transporters, which are labeled along the top of the table, and then the compounds are labeled along the side of the table. What we did was base our concentrations on clinical pharmacokinetic data for each inhibitor, using common dosing regimens in the clinic for CYP3A DDI studies, upwards of 10-fold higher than the average Cmax at steady state. So you can see some of them are highlighted in red. These are compounds where we did an initial range-finding IC50 experiment, and we needed to adjust concentrations lower as the transporter is quite sensitive, and we needed more points after the IC50. Here are the inhibitor concentrations we used for the vesicles. So again, the transporter is along the top and the compound is along the side. We utilized the same clinical PK data and DDI studies for these sets of assays, but only ritonavir needed to be decreased following an initial IC50 assay. So for all of our experiments, we used prototypical inhibitors that can be found in the literature and across the industry. This includes the use of rifampin for the OATPs, probenecid for the OATs, and quinidine for the OCTs. Cimetidine inhibits both MATE1 and MATE2K, while verapamil inhibits PGP. We use KO143 for BCRP and benzbromarone for MRP2 and 3. Finally, we utilize cyclosporin for BSEP inhibition. All of these data can be found in the supplemental information of the paper. I'll jump right into the data. As you can see from the previous tables, we had quite a few different graphs, so I will only show a couple today. You can find the IC50 graphs in the supplemental materials as well of the paper if you're interested. So we're looking here at inhibition of OATP1B1 by ritonavir. This is in our stably transfected HEC293 test system. Testing concentration range from 0.3 to 10 micromolar, we see a nice concentration-dependent decrease in the uptake of estradiol glucuronide into the OATP1B1 transfected cells. When we utilize GraphFit, we can find that we have an IC50 of 0.68 micromolar. The second example is inhibition of PGP by clarithromycin. This is using NMQ, or N-methylquinidine, and our membrane vesicle transfected with PGP. Again, this is an ATP-dependent test system. So when we increase the concentration of clarithromycin from 0.3 to 50, again, we see a nice concentration-dependent decrease in the ATP-dependent uptake of NMQ. Using GraphFit again, we see our IC50 is approximately 8.9 micromolar. This results table is shown in the paper, in the manuscript itself. It's all the IC50 values that we determined for each of the transporters. We'll just highlight a few things here. So the compounds that are highlighted in yellow inhibited the fewest transporters, and those include itraconazole, N-desalkyl-itraconazole, and clarithromycin. And the transporters that are shown in green were inhibited by the fewest compounds. So this includes MATE2K, which is only inhibited by ritonavir, MRP2 and MRP3, which did not have any inhibition at any of the concentrations that we tested. On the other hand, some transporters are more sensitive, including PGP, which demonstrated 50% or more inhibition by all of the compounds that we tested. Getting into the figures that we have in the manuscript, the first one that you see here is the DDI prediction for hepatic uptake and efflux transport based on the FDA's basic model. So we're looking here at I1 over IC50, or the total Cmax over the IC50. The cutoff for this from the FDA is 0.1. So any values greater than 0.1 indicate there could be a clinically relevant drug-drug interaction at that transporter. We can see that ketoconazole on the far left of the graph had a value that was 200-fold greater than the cutoff for OCT1. Ritonavir had, again, over 100-fold greater than the cutoff value for PGP. Itraconazole, as well for PGP, had over 200-fold greater than the cutoff. And hydroxy-itraconazole led to considerable inhibition of OCT1, which had a value that was 550-fold greater than the cutoff value. We also looked at the sum of itraconazole and its metabolites, and for PGP, BSEP, and OCT1, they all had considerable effects on those transporters. It's important to note that Key's paper says that the itraconazole metabolites can contribute to 40 to 50% of the overall impact on cytochrome P450 inhibition after multiple doses. So we can see here is there's the potential for this to have these compounds to also have an impact on drug transport as well. We saw, again, none of the MRPs were affected by any of the compounds that we tested. So we have to ask ourselves, what's the clinical relevance behind this data? So currently, there's not much clinical information regarding DDIs with drug transporters in the literature. We do know that verapamil can inhibit OCT1-mediated uptake of metformin with a calculated IC50 value of approximately 13. What's interesting is that it appears on the surface this would not actually cause a problem because plasma PK of metformin is not altered. But on the other hand, what we see is a major shift in the AUC of glucose. In fact, it decreases over 240% in the presence of verapamil. So these data demonstrate that while we might not always see a direct effect on the victim of the DDI, there can be downstream effects that lead to clinically relevant interactions. The next graph that we have here is looking at DDI predictions for OATP-mediated hepatic uptake, and this is using R values. And R value is defined as 1 plus the fraction unbound times the maximum concentration at the inlet to the liver over the IC50. The cutoff for this is 1.25, so any values greater than this would indicate a clinically the potential for a clinically relevant interaction at that drug transporter. We can see that both ketoconazole and clarithromycin were predicted to affect both OATP1B1 and 1B3 using the R value of greater than 1.25. On the other hand, ritonavir and itraconazole had values that were less than 1.25, and this also includes when you calculate itraconazole and the metabolites combined had values less than 1.25. Again, we need to ask ourselves, what's the clinical relevance behind this? So we know that ketoconazole co-dosed with bosentan leads to an increase in bosentan's AUC by over 100%. Furthermore, when clarithromycin is co-dosed with bosentan, glyburide, and pravastatin, we see an increase in their AUC as well. What we need to keep in mind is that OATP inhibition likely only explains part of these interactions, as we know that CYP3A is also inhibited by these compounds and could be playing a significant role. So it's important to note that all of these DDI interactions between the transporters, the victim drug, and the known inhibitor are complex. A number of processes are likely taking place here that need to be taken into account when we determine the best inhibitor for a clinical study. The third graph that we have here is the DDI predictions for renal and blood-brain barrier transporters, again using the FDA's basic model. This time we're looking at the unbound Cmax over the IC50. And again, the cutoff is 0.1, so any compounds with values greater than 0.1 are predicted to have a clinically relevant interaction with the transporter. So we saw that all of the compounds, all of the transporters had values greater than 0.1 for at least one of the compounds we tested, except for OAT1, which is not predicted to have any clinically relevant interactions. All values were less than 0.1. It's important to note all the values were still less than 10-fold higher than the cutoff of 0.1. We see that PGP and OCT1 were the most affected by the most compounds. We saw that ketoconazole and hydroxy-itraconazole had the greatest impacts on those drug transporters. And we also see that clarithromycin did not have any values that were greater than 0.1. So if we do a data search on the University of Washington's drug-drug interaction database, we can see that only MATE1 right now has a clinically reported interaction between ketoconazole and an antifungal drug that was being tested. So while the literature and current clinical data might suggest that MATE1 inhibition by ketoconazole is the only relevant one, it's likely due to a shortage in the knowledge about other drug transporters and how DDIs may affect their action, as well as the outcome on victim drugs. We can see from our data here that several of them have values that are considerably higher than the cutoff of 0.1, indicating there would likely be a clinically relevant interaction between the transporter and the drug that we are testing. The final graph here is looking at the DDI predictions for intestinal efflux. transporters based on the FDA's BASIC model. So this looks at I2 over IC50, and I2 is defined as the dose of the drug over 250 mils. The cutoff value is 10 here. Again, the three transporters we're most interested in for intestinal efflux are PGP, BCRP, and MRP2. We see again that MRP was not predicted to be affected by any of the inhibitors, as we saw no inhibition of that transporter by any of the compounds. On the other hand, for PGP, we see that our I2 over IC50 values can range anywhere from 30 to 1,000-fold the cutoff value for all four compounds that we're testing. Furthermore, for BCRP, the I2 over the IC50 values range from 8 to 30-fold the cutoff value for ketoconazole, ritonavir, and itraconazole, indicating we have some clinically relevant interaction that potentially could occur there. We see that clarithromycin was not predicted to inhibit intestinal BCRP. These are the most well-characterized drug transporters, and there's many reports of interactions between all of these inhibitors mentioned here, ketoconazole, clarithromycin, etc., and PGP substrates, including fexofenadine and quinidine, as well as BCRP interaction between rosuvastatin and ritonavir. So we do need to note some of the potential limitations of the study. There is the potential for nonspecific binding of the inhibitors. We know that these are highly protein bound in vivo. We did not directly measure this, and we asked ourselves whether or not we should. And the question we need to ask ourselves, if we try to calculate the free, or if we try to measure the free inhibitor concentration, should it be evaluated with or without the test system present? So if we have the cells or the vesicles present, then we needed to be able to determine how much was bound to the cell, how much was free inside the cell, and how much was bound to the test system. So the alternative was to employ pre-incubations. Because of these questions, then, we used a consistent design throughout every single assay that we ran. All of the compounds were pre-incubated for 15 minutes with either the HEC-293 or the vesicle test system. The inhibitor solution was removed, and then we added on our substrate. And in the substrate solutions, a fresh dose of the inhibitor was also included. So this study design was designed to use similar methodologies that are reported either in recent publications or what's utilized in industry currently. And what we wanted really to do is try to minimize the effects not only of time-dependent inhibition of the drug transporters by the compounds, but also this nonspecific binding problem that we might run into with some of these compounds. So, in summary, of all the alternatives to ketoconazole that we tested for strong clinical CYP3A potential inhibitors, the following conclusions can be made. We saw no inhibition of MRP2 or MRP3 by any of the compounds that we tested at clinically relevant concentrations. We also saw that MAY2K was only inhibited by ritonavir with a IC50 of 15 micromolar. We also saw that itraconazole, N-desalkyl-itraconazole, and clarithromycin inhibited the fewest transporters. And these include OCT1, PGP, BCRP, and BSEP for itraconazole, PGP for N-desalkyl-itraconazole, and OATP 1B1, 1B3, PGP, and BSEP for clarithromycin. If we look at the FDA's basic hepatic model, I1 over IC50, again, that's the total Cmax over the IC50, we saw that OCT1 was the most potently inhibited transporter. There were values that were 250-fold greater the cutoff of 0.1 for itraconazole and metabolites. We also saw that PGP was the most potently inhibited by ritonavir with an IC50 of 0.68 micromolar. And none of the inhibitors that we tested had I1 over IC50 values that were less than 0.1 for all transporters, except for the MRPs, which were not inhibited by any compounds that we tested. Looking next at the FDA's R-value hepatic model for 1B1 and 1B3. Again, an R-value is defined as 1 plus the fraction unbound times the maximum concentration at the inlet to the liver over the IC50. We see that ritonavir and itraconazole had values that were less than 1.25, indicating there is not the potential for a clinically relevant interaction. On the other hand, we saw that clarithromycin most potently inhibited 1B1 with an R-value of 3.5. If we look at the FDA's basic renal and blood-brain barrier model, the unbound Cmax over IC50, we see that OAT1 was predicted to be unaffected by all the inhibitors that we tested. We see that none of the unbound Cmax over IC50 values were greater than 10-fold the cutoff of 0.1. We also saw that hydroxy-itraconazole had the greatest impact. It inhibited both OATPs, OAT3, OCT1, PGP, and BCRP. We also saw that clarithromycin did not have any unbound Cmax over IC50 values that were greater than 0.1. And finally, looking at the FDA's basic intestinal model of I2 over IC50, again, the I2 is defined as the dose of the drug over 250 mils. MRP2 was predicted to be unaffected by all inhibitors, as this compound was, this drug transporter was not inhibited by any of the compounds we tested. On the other hand, we saw significant potential for interaction for PGP and BCRP by the inhibitors that we tested, with the exception of clarithromycin for BCRP. In conclusion, we can say that none of the alternatives to ketoconazole provided a clean inhibition profile towards all 13 drug transporters that we evaluated. So the alternatives to ketoconazole each have a unique drug transport inhibition profile. We saw that neither MRP2 or MRP3 were inhibited by any of the alternative compounds we tested. We saw that ritonavir and itraconazole may be the best alternatives for CYP3A substrates that are transported by 1B1 and 1B3, as these were not predicted to have a clinically relevant interaction between them. We also saw that CYP3A substrates that are transported by OAT1 may not be affected by any of these alternative inhibitors. None of the values were greater than 0.1 for this transporter. Clarithromycin may be your best choice for substrates of renal transporters. And for substrates of intestinal PGP or BCRP, clarithromycin again may be the best choice, although we have to keep in mind that PGP is still predicted to be affected by clarithromycin. So the overall goal of the paper was to take the FDA's basic static model to provide a clinically relevant context using concentrations that would be potentially present in vivo following a dose that's used in the clinic typically. It's important to keep in mind that each compound has a unique transport inhibition profile, and while all of the predicted potential interactions that we talked about today, including the R values and the I over the IC50, may not necessarily all be clinically relevant, the drug transport inhibition profile should most definitely be taken into account when you investigate a drug that may be a victim or a perpetrator of drug transport inhibition. It's also important to keep in mind that a typical drug clearance study for CYP3A doesn't usually take into account other clearance mechanisms. They're considered minimal unless they're reported otherwise. But it's important to know the PK of your drug, the pharmacokinetic parameters of your drug, and if drug transport plays a role, which may affect the outcome not only of drug metabolism but also of drug clearance. So overall, we state that the best choice for a strong clinical CYP3A inhibitor depends really on the unique transporter substrate profile of the drug candidate. So I also need to thank Andrea Wolf, the head of the drug transport department where this work was completed. I also need to thank Sekisui Xenotech's Analytical Services Group for their help with the NMQ analysis, and I'd like to thank all of you for listening. We'll have questions right after Matt goes through a few announcements.
Thank you, Lydia. As a reminder, if you have any questions regarding Lydia's presentation, please remember to type out your questions and send them in via the questions pane using the GoToWebinar user console. Before we move into Q&A, short mention about an upcoming show. We will be at AAPS Drug Transport Workshop in Baltimore next week, April 18th through the 20th. Dr. Vermeer will be there, as well as our technical account manager, Lindsay Harston. We will be at booth 4 as an exhibitor. Please do stop by, introduce yourself. If you have any questions for Dr. Vermeer regarding today's presentation, she'll be able to answer them. Also, we will be presenting a poster at this conference, poster M1032, which will be presented Tuesday, April 19th at 5:15 p.m. At the AAPS Drug Transport Workshop. This poster covers this publication. Lastly, as Lydia mentioned early on in her talk, she mentioned some of the transporters that are available from Sekisui Xenotech. That's just a small dose of what we have. We have over 50 transporter assays available at both our Tokai and Kansas sites. We have a variety of models available regarding cell lines, vesicles, transfected cells, oocytes, hepatocytes, and in animal models. And all of our drug transport services are available as either GLP or non-GLP. So with that, I'm going to move into the questions part of the presentation. I'm trying to bring these up here. Can you read that?
Sorry.
You're good.
Okay, so one of the first questions we have is I2. So what is I2? So I2 is defined as the dose of the drug over 250 mils. So this takes into account an oral dose of a drug and then the volume of the intestines that's typically used. Never heard of that.
Okay.
There is a question regarding, I'm going to butcher this probably, cabestat as a strong inhibitor. I can try to get back to you about that. I'm not familiar with that compound.
All right. Doesn't look like there's very many other questions. If you guys have questions, please get them in. Those are the only two that we have at this point. Lydia, good job on explaining everything. We'll give it another minute or so if people want to write up some questions. In the meantime, I will share some information with you all regarding if you have questions for us, please feel free, if you're located in North America, to contact us directly. We do have distribution throughout Europe with Taboo Bio, and then also with Japan with Sekisui. And their contact information is listed there. So if you have questions regarding any of our contract services or in vitro products or reagents, please do contact either us or our distributors. So let's see, for compounds evaluated prior to the IN. One question just came in. Okay, and so we've just been joined by Dr. Brian Ogilvie as well, who's going to answer a few questions. Dr. Ogilvie?
Sorry, I just came in. I saw the question about cabestat as a strong inhibitor, and sort of the impetus for this study was last year at the Marbach DDI meeting, and Alex Kay was sort of the inspiration for this. She had gone through a lot of the alternatives, and one of them was actually listed as cabestat because, of course, it was designed in essence to inactivate CYP3A4. However, there isn't a single drug formulation at this point that I know of, so it's not included. I think it would be a good alternative if it were a single formulation.
Okay. All right, guys, I think that covers it. So I'm going to wrap up today's session. Again, thank you for taking time out of your day to join us, and thank you to Lydia for her wonderful presentation. If you have any questions, please do feel free to contact us or any of our distributors. You can find all of our distribution information on our website, www.xenotech.com. Thanks again, and have a great rest of your day.